This working? Sounds like it. Okay, so we ended last time. I gave you a result with some sort of telegenic explanation of how to get it and said you can work through and confirm it. But the result turns out to be a very important one. We were looking at the combination of selection and mutation, and in particular what's called the mutation selection equilibrium. Properly, it's not really an equilibrium. It's more of a non-equilibrium steady state because there's a distribution of states and you sort of wander around in that distribution of states. But the term that's used by population geneticists is mutation selection equilibrium. And the idea here is we were selecting against an allele, which we made recessive, but it keeps reappearing due to mutation. And so we wonder, on average, how many do we expect to see in the population? And we got that by taking our equation for change, which included mutation and selection, and setting the change equal to 0. So we basically solve for an equilibrium. You can check that this equilibrium is stable. I'm not going to go through that, but I just want to remind you what the result was. I use lowercase p hat for the frequency of the allele at mutation selection equilibrium. And for a recessive allele, that came out to look like the square root of the rate at which there's mutation to that allele divided by the strength of selection against that allele. Yes?
Could you repeat what p hat means, the frequency of the allele?
This is the frequency. So in general, I'm taking p as a frequency of, I'm calling it the A1 allele in a population. I'm putting a hat over it to indicate this is the value at mutation selection equilibrium. So p is just measuring the frequency of an allele. It could be very different from this. But this is the expected frequency under, long-term expected frequency under a combination of mutation and selection. Does that make sense?
Okay, and another question, how realistic is this model in regards of one allele mutating to another? Like alleles exchanging states, kind of like that? Is that realistic?
Of mutation back and forth between two alleles?
Yeah.
Yeah. Here's the thing. We're setting this up as though there are only two alleles, right? Real loci have a lot of different alleles, but I just say there's A1 and A2, and I have mutation back and forth. The trick, if we're only interested in the frequency of one of the alleles, A1, we normally think of these, this is a sequence, a particular sequence at a locus, and this would be a different sequence. In fact, there's no reason I can't say A2 is everything that's not A1. So if there are 20 alleles, we can say there's A1 and there are all 20 others, right? And in which case, the frequency of A2 is just the frequency of something not an A1. So that's the trick you use to add more alleles to it. So really, what mattered in this was this mutation rate. I called it mu1 then, I dropped the 1 here, coming in. From all the others, at what rate is A1 appearing by mutation? Does that make sense? This is a common trick in population genetics. And I don't have time to go into the math of this, although it's of interest and it's very, very new math. But there's a reason that they like to do that. They like to make it binary, right? These are categorical variables. Categorical variables act in some ways different from regular variables that we're looking at. Categorical meaning there are a set number of states and it has to be in one of those states. If there are only 2 states, it's easy to deal with them. You just deal with the frequency of one of them and you're done. When you have 3 or more states, it becomes increasingly difficult using standard methods to deal with categorical variables. And so people like to knock it down to just 2. And so that's, I think, one of the reasons I didn't realize until I started trying to develop a different body of theory for change in protein sequence, where you've got 20, or if you're a bacteria, potentially 21 different amino acids that could be at each site. Then you can't, and you want to keep track of each one of them. So you don't just want to say, what's the change in the frequency of alanine at this site? You want to follow all of those categorical variables together. In that case, you have to use a really different approach. Okay, but if there are only 2 variables, it's no problem, you just pick one of them. So I think that's one of the reasons that population geneticists tend to do this. They said, well, let's just look at this one allele. So this was our result for the expected long-term frequency under mutation and selection. Now, here's the thing. What sort of real values do we expect for this? It sort of makes sense. It's not 0 because mutation is occurring and the allele keeps reappearing. But the expected frequency is divided by S, the selection against the allele, because that's how strongly it's being selected out. Okay? But consider a case, just to think about it. Let's say mu 10 to the minus 6. For an entire gene, when people would talk about alleles at a locus, this is a reasonable rate for a mutation rate. Okay? So we're not talking about a site. We're not saying the mutation rate for this particular site from an A to a G. We're taking over a gene, all mutations to a different allele. 10 to the minus 6 is a reasonable value for it. Say S is 10 to the minus 2. That might seem like really weak selection. It's not super weak selection. Selection is strong enough to drive evolution in the face of drift in a population of easily 1,000 individuals, which is a small population. Okay, so this is small but moderately weak selection. And when people measure selection, try to measure selection coefficients in natural populations, you often see things on the order of 10 to the minus 2. Okay. Well, in that case, what do we expect from this? We've got 10 to the 6 over 10 to the minus 6 over 10 to the minus 2, but we're taking the square root of it, right? And so we expect our p hat to be around 10 to the minus 2, right? 1 in 100. That's actually a fair amount. Think about even a moderate-sized population, again, say a population of 1,000 individuals, you'd expect about 10 copies of those genes floating around, of those alleles, I should say, floating around in the population. Go for a bigger population. I usually, when I'm lecturing on this, I teach at Texas Tech University. It's in Lubbock, Texas. Lubbock calls itself a city. Compared to Sao Paulo, it's about the size maybe of a neighborhood here. So if we use the city of this size with the population of the greater metropolitan area, not just Sao Paulo proper, it's in the range of 20 million or so for the greater metropolitan area, I think. That's 20 million diploid organisms. Each got 2 copies. So 40 million gene copies that we're looking at. 10 to the minus 2, we're on the order of 400,000 copies floating around in this large metropolitan area. So the point is, we expect an appreciable number of copies of even deleterious alleles floating around in natural populations. And even if I made this, say, 10 to the minus 1, which would be quite strong selection, in a population this size, there would still be a very large number of them out there floating around. Why does this matter? Well, if you go back into like the 1950s, coming into the early 1960s, one of the things that population geneticists were interested in is how much variation is there just standing in natural populations. And there was sort of a debate between different people. Some folks thought, probably not much. And that was based on the idea that selection eats up variation. So the image was when a mutation occurs, it's either adaptive and it sweeps through the population, or it's deleterious and it's wiped out. Okay? And if you impose selection on a population, you're eliminating variation at a fairly high rate. So selection should just sort of eat up variation in natural populations. Other people noticed that there seemed to be circumstantial evidence that there actually is a lot of variation. One of those pieces of circumstantial evidence is simply that if you take almost any organism from a natural population, take a group of them, and you select for a trait, say select for them to get bigger or smaller, they do right away. The first generation that you select on a trait, it starts changing. It's not like you're waiting a long time for a mutation to come along. That was suggestive that there was a lot of variation floating around. Another thing that people noticed is if you have organisms that are normally outcrossing, so individuals sexually reproducing and outcrossing, so they mate with somebody different, very unrelated to themselves, if you inbreed them, so if you do, say, full SIB matings, you see all sorts of new variation showing up that you didn't see before, right? And what does that suggest? That suggests that there were a bunch of rare recessive alleles that are usually masked in heterozygotes, but when you inbreed things, when you cross close relatives, you start to get homozygotes for those recessive alleles and you start to see them coming up. So those were the sort of circumstantial evidence that there was actually a lot of variation. There's a long history of how people evaluated it, but it turns out there is a lot of allelic variation in practically all natural populations. One sort of illustration of that is people use the term equivalent lethal units for deleterious alleles. So if we've got, say, in the human population, a bunch of recessive deleterious alleles. So they're only expressed when they're homozygotes, and they're rare, so they don't become homozygous very often. You can ask, how many of them would it take to kill you? And then how many, if you were homozygous at all those sites, and then how many of those does a typical person have? And it turns out a typical human has about 4 lethal units. So we have the equivalent of about 4 completely lethal alleles. They would be lethal if they were homozygous. But they're not homozygous. In the vast majority of people, they're heterozygous, so we don't see them. But they're floating around out there. Now, those are alleles that are strongly selected against. So S is large for those. Think about alleles that are only slightly selected against. They tend to, say an allele tends to make individuals a little bit smaller. Body mass is a little bit smaller. Not much. It's not very strong selection, but it tends to make you a little bit smaller than is optimal, and so it's selected against. You have a bunch of those alleles floating around in the natural population. Why is that important? Well, what happens if the environment changes and you select for smaller individuals? That genetic variation is already present in the population. You don't have to wait for a new mutation to come along. It's already there, they start increasing in frequency. And recombination kicks in. As these rare alleles that are already there, some of them start increasing in frequency, they start recombining, and so you quickly start getting new genetic variants showing up. As they increase, new combinations appear. And every time a new mutation appears, if it starts to increase, it doesn't create just one new variant. It creates all the possible combinations of it with other alleles at other loci in the population. Initially, it appears in one genotype, but if it starts to increase, it starts over time getting recombined with others and others and others. And so a lot of new variation is showing up. What this is getting at is the fact that for most natural populations, evolution is not mutation limited. Evolution is not mutation limited. By which I mean the rate of evolution is not determined by the rate of mutation. One reason I'm emphasizing it here to this audience, because y'all are doing mathematical biology, and one of the most common things I see when folks decide, hey, I'm going to model an evolutionary process, especially when physicists decide to model an evolutionary process, they are stuck on this. They imagine that a population is basically uniform. And then you throw in random mutations, and occasionally one of those is adaptive. And if it's adaptive, it instantly goes to fixation. and then you've got a uniform population, and you wait for the next adaptive mutation to appear. That's not how natural populations work. Okay, the variation to select on is already there in the population. And this is an important thing for understanding, first of all, why evolution happens so fast. If you change selection in a population, instantly, the next generation, it starts to change. This is a reason why. This combined with recombination is a reason why if you keep selecting on a trait, it keeps changing. There's a selection experiment going on in the state of Indiana in the United States. It started in the late 19th century, I think in the 1880s, and then they really started being careful about it every year in the 1890s, selecting on corn. Corn is an economically important crop. A lot of it's grown in Indiana, so they decided to do an experiment where they just picked different traits of corn that were economically important. So things like the amount of oil in a kernel of corn, the amount of sugar in a kernel of corn, so forth. Those were the main things, because those are the main things that are economically important. And they said, okay, let's just start selecting on these and see what happens. Of course, you select on it, it changes. But they kept doing it. They're still doing it. You can go to their website and you can download the data. They stopped for a few years during World War II. And so if you look at the data, there's about a 4-year block where nothing changes. And what they did is, corn is an annual, right? So it grows up, they collect the seeds, the next year they plant the next generation. So generation time is 1 year. They just saved the seeds for a few years and stopped doing the experiment because everybody else was dealing with World War II, and then they started again. So there's this little blip in the middle. But otherwise, if you look at it, the change is essentially linear over now more than 100 years. It's just like that. That keeps changing. And one of the things that they wondered about was how far can we select it before we run out of variation? They never run out of variation, right? And that surprised people that they didn't run out of variation. The reason is that initially there was a lot of variation due to mutation selection equilibrium. As they started selecting, rare alleles started appearing in new combinations by recombination. And over the generations, sometimes new mutations do come in, but one new mutation gives you a huge amount of new potential variation because you have all the combinations that that mutation could exist in with other alleles at other loci. And so essentially the change is just steady. And if you look at the mean, I pulled out the data for oil content, the mean oil content in 2004, when I pulled it out one time for a lecture, it was about 4.5 times as large as it was in 1899 when they started really consistently doing this every year. And it's still changing in the same direction. And the distribution is overlapping. So if you look at 1899, you look at oil content, 1899 you've got a distribution over here, and 2004 you've got a distribution over here. Right, so it's changed immensely in that time, and it's still going. So in other words, evolution does not involve sitting around waiting for a new mutation, then a big change, then wait for another mutation. Even though that's one of the most common ways that people model it, and it's a common assumption that a lot of people have about the process.
What's the name of the data set, or the experiment, or how do you look at it?
I think it's the Indiana corn experiment. If you send me an email, I can get you, if you Google that, you'll probably find it. And if you send me an email, I have a link to the experiment. But do Indiana corn experiment, you'll probably come up with it. And you know, it's not the best website in the world, but you hunt around, you find where you can download the data. So, does this make sense? The importance of mutation selection balance is maintaining a lot of the variation that we see in natural populations. And when people realized there was a lot of variation, they started wondering what is maintaining it. We talked about balancing selection the other day that maintains 2 alleles in a population at the same time. One idea was maybe there's a lot of balancing selection. It turns out that's not maintaining most of the variation. There are a number of known cases of balancing selection, but it's not nearly enough to explain how much variation we actually see. It's mutation, selection, equilibrium that's explaining what we see. Incidentally, this was for a recessive allele. If we did it for a dominant allele, we end up with simply mu over s, not the square root of mu over s. You can see that makes a big difference because we're dealing with a number that's fairly small, much less than 1. Take the square root of it, you increase it by quite a lot. And this is consistent with what we see in natural populations. Deleterious dominant alleles are out there in natural populations, but they're far, far fewer than deleterious recessive alleles. Most of the variation is recessive. And this comes up in things like studying speciation. When we look at the alleles that when they appear in a population reduce the fitness of hybrids with another population, this is something people study a lot in terms of understanding how speciation occurs. Most of those are recessive. The overwhelming majority of them are. Does that make sense? Yes. Okay, so, yes, question?
Professor, could you talk more about which are reasonable values for S?
Yeah. So when people try to estimate selection coefficients in natural populations, they usually come up with, it seems to me, there's a distribution of values, and of course the weaker it gets, the harder it is to detect. So the stronger selection, the easier it is to see because you need a smaller population, a smaller sample to detect it. Even with that, 10 to the minus 1 is quite strong. And one study, Endler wrote a book, this was way back, so it's using old-fashioned methods, but they found very few cases of selection stronger than 0.1. There were a few that were up or around S of 0.5. Most were in the range 10 to the minus 2 to 10 to the minus 1. So that was sort of the range they saw. That said, we know sometimes really strong selection happens in nature. And what they were doing was just sort of picking a natural population that's sitting there and saying, okay, what do we see going on in it in terms of changes in allele frequency right now when we just happen to pick an interval and we decide to look at it? You're not likely to catch the really extreme selection events when that happens. There have been cases where people have caught very strong selection in action, and it can be quite strong and evolution can occur quite fast. The most famous ones was from guppies in Trinidad, an island off the north coast of South America, and it's kind of mountainous, and there are guppies, little fish, that live in streams coming down out of the mountains. And it's an easy system to manipulate because you've got these streams coming down and there are ponds and there are guppies in the ponds. And these guppies are not like salmon, they can't go upstream very well. So if you go up high, you'll find ponds with no guppies in them. But a little bit lower, there are ponds with guppies. It's not very far away, but the guppies just can't get up that waterfall. Guppies are like that long. And so you could do experiments. You could take some guppies from down here and put them in a pond up here and watch what happens. And in particular, the guppies living in ponds where they had predators, they did an experiment where they just took them and put them in other ponds without predators. And then they watched them change over time. And they also did this in the lab. They brought them into the lab and they had them in tanks with and without predators. And basically, the predators on guppies are other fish. They are visual. So when they see, they're detecting guppies, their prey, by visual cues. In the absence of predators, male guppies tend to have a lot of red on them, right, for sexual selection to attract females. When there are a lot of predators around, they don't have so much red because the red makes you stand out and predators see you. Also, when predators are around, the guppies develop much more quickly. They grow faster, their age of first reproduction is earlier, and they end up smaller. And this makes sense because there are predators out there who are likely to eat you. You better get to the age of reproduction and reproduce as fast as you can before you get eaten. Whereas when there are no predators around, they tend to live longer and reproduce and spread out reproduction more. And these are genetic changes. People have tested it in the lab. It's not a phenotypic plasticity case. It's a case for its actual genetic differences. So people could measure how fast do these traits like body size, red coloration, time of reproduction, how fast do those change in when you take away predators. Or conversely, they did the reverse experiment if you take something with no predators and you put it with predators. And turns out quite fast. They looked over about 11 years, which is about 22 guppy generations. And to give an idea of how different this was from, they changed, by the way, about 20%. Most of these traits changed by about 20% in that time, okay? The change is enough that I'm not a fish expert. I don't study guppies. I can look at the two cases and easily see this one has predators, this one doesn't, right? So it's not very subtle differences, which wouldn't be subtle to a guppy, but even subtle to a human. It's quite substantial changes. They looked at the total rate of change, and to give an idea of how this compares to what people used to think the rate of change in evolution was, if you go back into the middle 20th century, a guy named J.B.S. Haldane came up with a measure of the rate of evolution. And he called it the Darwin, so it's measuring rate of evolution. And the unit he identified was a Darwin, who's designated lowercase d. And this was a change by a factor of E in 1 million years. Change by a factor of E in 10 to the 6th years. You can guess that this involved taking a ratio of values and taking the log of that ratio. That's where the E comes from. And why 10 to the 6th years? I don't know, because J.B.S. Haldane was a geneticist, and a geneticist should have done this using generation time, not clock time, but this is what he did. Anyway, he looked at the literature in paleontology and concluded from that literature, he took where people had measured what looked like fast evolution, and he calculated how many Darwins it involved. And he came up with fast evolution in the fossil record looked like around 3 to 4 Darwins. So thought that fast evolution was 3 to 4 Darwins. And so we thought, okay, this is a reasonable measure. You know, 1 Darwin is sort of moderate evolutionary rate. And this was estimated mainly, the fast values were from a sequence of horse fossils. Horses got big pretty fast, right? You go quite a ways back, Heracitherium was about like this, small creature, and they got big rather fast. And so they thought, okay, that's really fast evolution, 3 to 4 Darwins. Okay. This guppy example I mentioned, they took their data and they said, okay, let's see how many Darwins it was. Most people don't use Darwins now, but they knew about this. How many Darwins? It was more than 3 or 4. It was 40,000 Darwins is the rate of change that they observed in that guppy population. Okay, and this has been done other times. Famous example, I won't go through it, but Finch beaks in the Galapagos Islands when there was a drought that changed the distribution of seeds that the finches had to eat. The beak size changed at a rate of about 20,000 Darwins over just a few generations. So the rate that evolution can occur in natural populations is 4 orders of magnitude larger than what people thought fast evolution was based on the fossil record. And one of the reasons is that natural populations have a lot of variation already there. And so if you select on it, if you do really strong selection, it changes really fast. And just so, you know, why did people come up with that low rate from the fossil record? Think what they're doing with that. They're looking at their horses. Time is here, and now time is geologic time, okay? And they're looking at, say, horse body size. And they've got a few fossils. The fossils are spread out like that over time. Each fossil is just a point, and fossils are usually at very different times. And so they're taking this long-term trend and they're taking the slope and they're saying, okay, that's the rate over time. In fact, this population may have been going like that, right? There could be very rapid change. You wouldn't see it in the fossil record. If you took the guppy example, for instance, in the fossil record, it would just look like, the research was done over 11 years. You can't distinguish 11 years in the fossil record. So it would just look like two values at the same point. And they say, oh, it was just variation, two different species of guppies. No, it was the same species of guppies. These 11 years later had been in a pond without predators than those had. But that's something you can't tell looking at fossils. So estimates of rates of evolution from the fossil record are literally many orders of magnitude lower than the rate at which it actually occurs. That makes sense? That's sort of a digression, but it connects to this issue of there being a lot of variation in natural populations. So we've been talking about population genetics, and population genetics as a field is traditionally identified as the change in the study of the change in the frequency of alleles in a population, okay? And we got those population genetics equations from the Price equation by putting in certain assumptions, the assumptions, standard assumptions of population genetics. Another field, and I'm just going to briefly touch on this because of lack of time, but another field some of you know, somebody asked about the other day, is quantitative genetics. And this is often treated as distinct from population genetics. Here, we're looking at continuous traits. So continuous traits that are influenced by many loci. So things like body mass, basal metabolic rate, all sorts of traits that you could talk about. Most traits that we talk about for organisms are influenced by a lot of different loci, okay? And so quantitative genetics focuses on that, and because it's assumed that there are a ton of loci and you don't know the details about what any of them are doing, you have to sort of summarize all the effects of genetics. And the way you do that in quantitative genetics, how do you quantify transmission across a generation? In population genetics, that's easy. You're talking about alleles. And so we assume Mendelian transmission of alleles. Okay. Quantitative genetics, you're dealing with some large number of loci, each of which may have some large number of alleles, and you don't know how many loci there are or how many alleles at each locus or which ones there are. Okay. What you can do is look at parent phenotype and offspring phenotype and see how those are related to one another. And so in quantitative genetics, you look at that, and if you look at the plot, the phenotypes of offspring as a function of the phenotypes of their parents, you often see something kind of like this. There's a lot of variation for a particular range of parent phenotype values. There's a lot of variation in offspring. But you can sort of capture the relationship by taking the regression of offspring on parents. Okay. That regression, the slope of that regression, is called the heritability. And it is designated little h squared. Purely historical accident or just historical artifact that's designated little h squared. Quantitative geneticists never talk about little h by itself. It's always h squared. Okay. Yeah.
How do we do when we have sexual reproduction? So we have two parents. How do?
Generally, in that case, you take what's called the mid-parent value. Mom plus dad divided by 2. That's what they do. And those folks in Indiana who are checking corn, they're quantitative geneticists, right? They're interested in this trait, how much oil there is in corn kernels. They care about it because it affects how much money people growing corn can make. And so it's an important trait. It's obviously controlled by many alleles. So they're doing things like estimating the heritability. Incidentally, heritabilities almost always are less than 1. And I won't go into, we can talk afterwards if you're interested about why that is. There's a straightforward reason why, but it's a little complicated. But I'll note, in general, h squared is less than 1. I've used the term regression here. Did that term sound familiar when we talked about statistics? Regression? Okay, everybody knows that term. This is where the term regression came from. Y'all may know this, but I did not know this until I was in graduate school. And in my PhD committee, the way our program worked, I assume it's similar here. At some point after you've been there a couple of years, you do a qualifying exam where you meet with your committee and they give you a bunch of written questions. You answer them over a period of weeks, and then you meet with them for an afternoon and they just throw questions at you. And you have to answer their questions satisfactorily to advance. And when I did my qualifying exam, there was only one question I just totally missed. I had no idea. And it had to do with regressions. And one of my professors asked me about regression. He says, why is it called that? And I started saying, oh, here's how it works. And he said, no, I know how it works. Why is it called a regression? I had no idea why it was called a regression. Y'all may know this. It comes from actually estimating exactly this, heritability. And the reason, and the fact that heritability tends to be less than 1. And the reason is that if you go back into the late 19th century and early 20th century, when people were developing these sort of statistical notions, people like Pearson and those guys, there was this thing folks noticed when they were breeding animals and plants. Say you've got your cattle and you want to select for higher milk production. Well, you select the cows that produce the most milk, okay? And they differ by the mean milk production by some amount. So say the cows you select produce 10% more milk than the average cow in the population that you have right now. And then you breed those cows, you get the next generation. Well, the next generation produces more milk, right? But not 10% more. It maybe only produces 5% more milk. In other words, you selected on a trait and it changed in the direction you wanted it to, but not as much as you thought it would. It doesn't change as much as you thought it would because heritability is less than 1. Getting more milk from your cows, if you're a dairy farmer, is progress. What it looked like is the trait was regressing back to where it had been. In other words, you're selecting for it to go this way, and it is going that way, but it's like it's always pulled back a bit, regressing. And hence the old term, at the time they called it regression to the mean. This is where that term came from. Now regression is used, just a mathematical term, all over the place, and nobody thinks of that. I'm just telling you, in case anybody ever asks you that, you'll have the answer. I did not, and like I say, it was, I still passed the exam, but it was the only one, it was just, I just had to say, I don't know. I have no idea. And then they explained that. Incidentally, sometimes you see it called regression to the mean. I saw an old paper by Carl Pearson, one of the founders of statistics. He called it regression towards mediocrity, which is a very value-laden kind of thing. It's like, you're trying to get superior things, but no, they are regressing towards mediocrity. Nobody uses that kind of nomenclature anymore. Anyway, so heritability is measured as a regression, hence the original term. And in quantitative genetics, what you're doing is you're essentially assuming that you can write offspring phenotype, expected offspring phenotype, as a function of parent phenotype and that slope of that regression. And so let's say we want to apply the price equation to this. So remember our price equation was delta phi bar was 1 over W bar, covariance phi offspring with W, plus this delta bar term. How do we get phi offspring? Well, if you make the quantitative genetic assumptions, phi offspring is equal to the mean phenotype in the population plus heritability times the phenotype of parents minus the mean. Right, so basically you're moving your axes to here, to the mean of this population. And in the absence of selection, so they bring selection in separately, but in the absence of selection, you expect offspring to be that mean plus how far your parents are from the mean times the slope of the line. Okay. So what if we just take that and we plug it in here? We get 1 over W bar covariance of, I'll write that, phi bar plus heritability times phi minus phi bar, that with fitness. Here's a nice thing. First of all, if you're not familiar with this about covariances, the covariance of A plus B with C is equal to the covariance of A with C plus the covariance of B with C, so it's linear in that sense. Also, if K is a constant, the covariance of A with K, whatever A is 0. So the covariance of anything with a constant is 0. Okay. Apply that here. Mean phenotype is a constant in the population. It's a property of the whole population. Everyone has the same mean phenotype. If we took, say, the phenotype was height of people in this room, there's a single mean height of people in this room. I have a height. In a sense, I have a mean height for the room, but my mean height for the room is the same as all of your mean heights for the room, because that's just a property of the whole room, right? So that's a constant. That's a constant there. Heritability is a constant, right? Because it's a regression over the whole population. So really, this breaks down to 1 over W bar covariance of, the only thing we have left is heritability times phi, h squared phi with W. And because heritability is a constant, it can come out. So I can write it as h squared times 1 over W covariance phenotype with fitness. Now, originally, this is offspring phenotype. But now we've got covariance of parent phenotype with fitness, as though there's no mutation, no recombination, nothing else is changing. This is just the mean change just due to selection. So it's the difference between the mean of parents after selection and the mean of parents before selection. Okay? And in quantitative genetics, this is called the selection differential. And designated capital S. Yeah.
Is that W bar, 1 over W bar?
Yes.
Okay.
Sorry. Important. That term will become really important in about 5 minutes. Right now, it's just a constant that's there. So this is called S, the selection differential. In other words, basically, this is how much selection is changing it. But then when you produce the next generation, how different are they from the current generation? That, in the way we're writing it, that would be delta phi bar. In quantitative genetics, that's called the response to selection. And when you put these together, this is designated capital R. We could write this as capital R, response to selection, is heritability times the selection differential. If you are a biologist and you've taken a quantitative genetics class, you've seen that. That's called the breeder's equation. That's sort of the central equation in quantitative genetics. Quantitative genetics is usually treated as this totally different subject from population genetics. It makes different assumptions. We got their main equation from exactly the same place that we got our population genetic equations, from the Price equation. Traditionally, this is derived completely differently from it. And I'm using this to drive home that part of the value of this Price equation is that because it's a general rule, you want to get population genetics, put those assumptions in. They assume random mating, right? Sometimes they assume no mutation, okay? Random mating, no mutation. They're talking about alleles. It's either that allele or it's not, so your phenotype 0 or 1. Plug those in, you get population genetics. Quantitative genetics, you're talking about a continuous trait, and you're paying particular attention to the regression of offspring on parents. This distribution is assumed to be a normal distribution in quantitative genetics. Well, plug those assumptions in, you get quantitative genetics from it. Yeah.
What's the name of the equation again?
What's that?
The name of the equation
The
again?
name, oh, yes. This is called the Breeder's equation. And you can guess, quantitative genetics is a very applied subject. I told you about that corn experiment. That's classic quantitative genetics. It originally came from agriculture. And so they're not concerned with understanding fundamental evolutionary principles, although quantitative genetics has yielded a lot of insights about evolution. They're mainly concerned with how do I get more oil out of my corn, how do I get more milk from my cattle. And you can tell that applied quality because it has terms like the Breeder's equation. There's a term that is used in quantitative genetics also that you assign to an individual called the breeding value of that individual, which seems odd if you're just thinking of evolution in general, but if you're thinking of, in particular, agriculture, that makes sense. It's like this bull has a high breeding value. That means it's valuable to a person who is an agriculturalist. Anyway, I'm not going to dwell on that, but I want to show that you can get sort of the main results from these different approaches, what we're treated as completely different approaches to evolutionary biology, from our same initial result here. In a sense, the price equation is playing the kind of role, it's not the same as, it's not a conservation law, but it's similar to the principle of conservation of energy, or conservation of matter energy in physics. If you've done some physics, you know you can derive a lot from that, right? You can derive the Schrodinger equation mainly from conservation of matter energy. And so this sort of basic rule that you say this has to apply everywhere, you can use to derive all sorts of different cases. The price equation is playing a role like that in evolutionary biology. Does that make sense? Okay, so now what I want to do is talk about something that's left out of this. I said it's always true, and it is, but there's this little catch. Prior to production of offspring, how do we know what W is, and how do we know what phi offspring is? This is an exact statement if you look after the fact. If you let a generation occur, and then you say, okay, what were the phenotypes of the descendants, and what were the fitnesses of the parents? You plug it in, this will always be true. But say we're looking forward a generation. In that case, we don't know exactly what W or offspring phenotype are going to be. Okay? So we want to modify this. We want to make this stochastic. In particular, we want to derive a stochastic price equation. Because this is what we'll want to look forward. There might be an expectation. I honestly had an expectation that what you would get is something that looked just like the price equation, but maybe you had expected values in front of some of the variables. You get that, but you get some other things too. New terms show up, including new directional evolutionary terms, new terms that drive directional evolution when we make this stochastic. Now, the approach that I'm going to use is a little bit different from what you usually do in stochastic process theory or stochastic differential equations. I know you guys had a session on this before. I don't know how it was done there. Often, Stochasticity is introduced to traditional models by adding an extra variable. So if we wanted to make fitness stochastic, we might have W plus another variable, right? Does that sound familiar? So say I had W plus, call it DW. DW is a bad example. SW. And then say this is a random variable, right? And then go through my calculations with this new thing. Does that look familiar as a way to do stochastic process? Often also when people do this at this stage, they'll put some restrictions on the distribution of that. They'll say, okay, it's normal 0, 1, right? Normally distributed, mean of 0, and variance of 1. That's a common trick. We're not going to do that. Okay, so we're going to take a totally different approach to making this stochastic. What we are going to do is simply state that W and offspring phenotype and other things in more complicated cases, but we're just going to focus on these. We're going to say these are random variables. And random variable here means it has a distribution of possible values. We don't characterize it just with a number, we characterize it with a distribution. Okay. Conceivably, that could be a Dirac delta function as the distribution, but we're not going to be using that case. It's always going to be the case that we're looking where these can take multiple values. Okay. So, in other words, random variables are characterized by a distribution of possible values. And we're not going to make any assumptions about the shape of those distributions because biologically, you don't know what the shape of the distributions will be. I mentioned that people often assume things are normal, the variance is normal 0, 1. Some of the early, and even now it's common, stochastic population genetics does that. They add a variable and they assume that it's normal 0, 1, and they do that with fitness. And this always gets me because I mentioned this the other day, the normal distribution is defined as minus infinity to infinity, right? That means there's a certain probability that the organism has -3 offspring, which is biologically meaningless. Okay, they get around that problem by assuming the variance is very small. And so then you kind of ignore those negative offspring and it doesn't really mess up your calculations very much. We're not going to do that. We're going to allow any distribution possible. Something else I'll say about the use of the term random variable. I will use it only for something that is truly a probabilistic variable. Okay? So when we're talking about phenotype, I say offspring phenotype is a random variable. I'm going to say phenotype of parents is not, okay, because it's just there. If our phenotype is height, my height is my height, okay? It's just here. I'm ignoring quantum variation in height. Basically, it's just a value. The height of my eldest son when he gets to be the age that I was when he was born, that's a random variable. Okay, I don't know exactly what that'll be. I can make a reasonable guess because he's 19 now and he's probably not going to grow a whole lot more, but you never know. People can change, right? So that's uncertain. So when I say something's a random variable, I mean it really is uncertain. In statistics, the term random variable is used sometimes in a slightly different way. So a statistician might be fine saying, okay, the height of a person from this room is a random variable. Right? I would say, no, it's not. Right? Your height is your height, your height is your height, your height is your height, right? But a statistician, when they say that, here's what they mean. They mean if we pick a random person from this room, what's their height? That's a random variable, but the randomness comes in, the uncertainty comes in because they're taking a random sample. When we're doing stuff with the price equation, the means and variances and covariances and things that we dealt with in a population are over the entire population, not taking a sample. So parent phenotype, things like that, are not random variables. Okay? Offspring phenotype is a random variable. Fitness is a random variable because before reproduction, you don't know how many descendants an individual is going to leave. Okay? Another one, mean fitness, W bar. That's the average in a population. But remember, each individual's fitness is a random variable. That means the mean fitness in the population is also a random variable from a probabilistic standpoint. We don't know exactly what it will be. Okay? So if we're going to do this, we're going to have to talk about means, variances, covariances, and things between these random variables in terms of their probability distributions. But we're already talking about means and variances and covariances within a population. Okay? This is why I used that notation that I did. So I'm going to distinguish frequency operations are the kinds of operations that we've been doing. Things like phi bar, variance in phenotype, covariance phenotype with fitness, with these square brackets. That means these are over a population of things that are there right now. Okay, so frequency operations are over a set of things with determinate values. We're going to have to also now talk about probability operations. I will use a similar notation, but instead of straight lines, I'll use angle lines. So offspring phenotype hat is the expected offspring phenotype. Offspring phenotype. As a probability operation. Okay, W hat is expected fitness. So I want to talk about the variance in fitness. Use angles. W, that is probability variance in fitness. We had, in the price equation, a covariance between offspring phenotype and fitness. Offspring phenotype and fitness are both random variables, probability random variables, so there can also be a probabilistic covariance between them, separate from the covariance within a population. I'll designate that like this. Again, angle brackets, phi offspring with fitness. What's the difference? If we go to a frequency operation, and I say, I talk about the frequency covariance of offspring phenotype with fitness, I mean over the population of individuals. This is over a population, okay? So this is a property of a whole population, okay? This is a property of an individual, right? And it might be zero, but it might be non-zero. If it's non-zero, it means for example, if it's positive, then if this individual happens to leave more offspring than expected, they will tend to have a higher phenotypic value than expected. So it's a relationship between your fitness and the phenotype of your offspring. Whereas this is a relationship between offspring phenotype in the population and fitness in a population. That makes sense? I'll give some examples going through doing this. But this is one of the reasons, aside from simplifying or using less chalk, to use this notation, because in fact there are two kinds of means, two kinds of variances, two kinds of covariances that we have to use if we're going to do this. If we're going to make this stochastic by just treating some of the terms as random variables. Anybody, any of the mathematicians ever study free probability? You see that? It's come up fairly recently in the past few decades in probability theory. And it's mainly concerned with non-commutative probability operations, where the expected value of A times B is not the same as the expected value of B times A. But one of the nice things about it is it makes it easy to talk about different kinds of expected values. Traditional probability theory, it's kind of hard to talk about that. In free probability, it's easy to. We are dealing with a case where there are different kinds of expected values. In traditional probability theory, that would mean you'd have to define separate state spaces and separate sigma algebras for each of those if you were doing it formally. And that gets kind of awkward. So, but there are some recent developments in pure probability theory that connect this approach to the pure math a bit better. But so the point is there are these two kinds of things. Now, here's the thing. Some things are both frequency variables and probability variables, right? So we have to be able to combine them. And to do that, I'm going to just give you a couple of important relationships for relating things. So the first one, something can have a frequency mean and a probability mean. Let's take fitness, for example. There's a frequency mean fitness in the population. It's the average fitness of everybody in the population. But that's also a random variable. Every individual has a probability mean fitness. And we could take the frequency mean of that. So we can take the frequency mean of the probability mean or the probability mean of the frequency mean. First principle that I'll give you, or a rule, is simple. Those are commutative. The probability mean of the frequency mean is the same as the frequency mean of the probability mean. So you can switch those. That should make sense because the mean is a linear operation. You add everything up and then you divide. But because it's a linear operation, you can just flip the order just fine. Taking the expected value of a variance or covariance, though, is a bit different. And in the price equation, we have a frequency covariance between two things. We're going to have to take the expected value of that. Okay? That's a bit more complicated. Here's a rule for that. If I use x and y, frequency covariance between x and y, if I take the expected value, the probability mean of the frequency covariance between two things, minus the frequency covariance of the probability means of x and y is equal to the probability covariance of x and y averaged over the entire population minus the probability covariance between frequency mean of x and frequency mean of y. I could show you how to derive these, but I'm not going to go through it here. But we need these rules. It turns out these are the only two we're going to need to make the price equation stochastic. Okay? So with these in mind, and by the way, this one, I tend to remember it this way better because it's square brackets with the angle hat out and in, equals angle brackets with the straight line out minus in. So it's easy for me to remember that form. What we're going to want to use, though, we're going to want to be finding this. So equivalent is x and y hat equals, I can just subtract that or add that to both sides, covariance of x hat, y hat, plus probability covariance x and y bar minus probability covariance of x bar, y bar. So this is equivalent. I just find it easier to remember the rule from that. And then whichever one I'm trying to find, I rewrite it. Okay? So why am I doing this? Because with these rules, we can very quickly derive a stochastic price equation. Let's write the deterministic price equation. Delta phi bar was equal to 1 over W bar, covariance, offspring phenotype and fitness, plus delta bar. In a stochastic case, there's not one change, there's a distribution of possible changes. To start out with, let's say, what's the expected change? Okay? To find that, I take the expected value of both sides, the probability expected value. Put hats over it. And then we use these rules to work with that. Now, here's one thing that turns out to be really important in terms of biological outcomes. You notice I put this hat over the whole thing because these terms are multiplied together. The expected value, either probability or frequency, of a sum of things is equal to the sum of the expected values. The expected value of a product is not equal to the product of the expected values. Okay? So I have to put a hat over both things. And this gives a hint of taking this approach rather than the traditional approach of adding new variables and doing stochastic process. What you do is just say, okay, I decree that these things are random variables. Then I start taking expected values, but you have to be careful, right? If you've got a product, you have to take the expected value of the whole product, not just the pieces. But when there's a sum, then I can take the expected value of the two. Here's something that you can see right up front is going to be confusing. I've got 3 different random variables in here, W bar, phi offspring, and W bar is in a denominator. I'm going to have to take the expected value of 1 over something. This is going to be complicated. Note, though, everything in here, if we just think of them as the frequency operations under this hat, W bar is a constant, a frequency constant for the population. It's the same for everyone in the population, right? It's a probability random variable, but as a frequency variable, it's a constant over a population. So I could put it anywhere inside. I could multiply it times either of these terms inside the covariance. I could multiply it by this. I could write phi offspring over W bar. That just gets confusing. Or I can multiply it by this, and I have individual fitness divided by mean population fitness. That sounds a lot more like something that's biologically meaningful. So I'm going to write this like this. The covariance of offspring phenotype with W over W bar. And we'll have to take the hat of that. So I've now moved that in. And I mentioned this, I sort of hinted at this before when we were doing deterministic models. You always have a 1 over W bar out front. And population geneticists and quantitative geneticists just tend to ignore it completely. What they do is they scale fitness so it's 1, and then you're fine. You know. When we make it stochastic, we can't do that because it's a random variable. Even if I scaled fitness so it had an expected value of 1, that wouldn't make this disappear because I'm taking the expected value of these things. So now I really have to pay attention to mean population fitness. So much so that basically, as we go through, we're constantly going to have a W over W bar term. So I'm going to define a symbol, omega, as a property of an individual. Omega of I is Wi over W bar. So properly, and this W over W bar does come up in quantitative genetics sometimes, and they call it relative fitness. So this is equal to relative fitness. The term relative fitness in intro textbooks and intro biology textbooks and intro evolution textbooks is often used in a different way, that just scaling it some way that's convenient. In the literature in population and quantitative genetics, relative fitness always means this, W over W bar. Okay. So, in fact, I'm going to rewrite this as omega. Delta phi bar hat. Now, how to deal with this? Well, we've got a rule for expanding the expected value of a covariance. So this is equal to frequency covariance of expected offspring phenotype with expected relative fitness plus probability covariance of offspring phenotype with relative fitness averaged over the population minus probability covariance of frequency mean offspring phenotype with frequency mean relative fitness. So I just applied this rule in this form to that, to expand it. And one of these terms is easy to deal with. Remember, omega is W over W bar. What happens when you take the frequency mean of it? Well, W bar is a constant, so it just sits there. The frequency mean of omega is the frequency mean of W divided by the frequency mean of W, which is 1, is a constant, always. And because that's always a constant, the covariance of anything with a constant is 0, so this is 0. And so we end up with expected change in mean phenotype is equal to covariance of expected offspring phenotype with expected relative fitness plus probability covariance of offspring phenotype with omega, with relative fitness, averaged over the population, plus this delta bar hat. The expected mean change just due to things like mutation that are happening in reproduction. This is our stochastic price equation. And the thing to note, it kind of looks like the original one that we had here, if I took the hats off of that. This first term looks sort of like what you might expect. It's got these things, but with just the frequency covariance with hats over it. But it's also got this whole new term sitting here. And in fact, there's also another novelty here because this omega, especially omega hat, expected relative fitness is going to behave in odd ways. Want to just first point out, so what's new here? This term. This is the most obvious one. This is fairly straightforward. What does this refer to? It's saying this, the probability covariance of offspring phenotype with relative fitness. What does that mean? Well, it means if your relative fitness ends up being different than expected, your offspring phenotype is likely to be different than expected. What could make that non-zero? And notice, so that's, without the bar, that's a property of an individual averaged over the whole population. What can make that non-zero? Well, one example, so I'm talking about that term now. One thing that can do that is what's called clutch size, offspring size trade-off. So I'll say example. Clutch size, offspring size trade-off. This is a term especially used in people studying birds. A lot of birds, most of them, build a nest and the parents provision the young, right? And so the parents are going out and collecting food and bringing it back for the young. And depending, sometimes one parent does this, sometimes both parents do it, sometimes one parent stays with the young and protects them while the other one goes out and gets food and comes back. But the point is the parents are actively working to bring food to their young. If they have a big clutch, if they have a lot of chicks in their nest, each chick on average gets less food. And because they get less food, they tend to grow up smaller. So pairs of birds that have a lot of chicks in one particular year, those chicks tend to be smaller because each one gets less food. If they only have a few chicks, the chicks can be bigger. That's the trade-off that's observed. And that is exactly what this is capturing. It's saying if you're lucky and you get a bunch of chicks in your nest, so this is large, then if phenotype is body mass, it's going to tend to be smaller. So for a clutch size, offspring size trade-off, this term will be negative, right? So it's capturing the effect of reproduction itself, the effect of how many offspring there are on their phenotype. Another case where this also shows up as being non-zero is something that I only realized fairly recently, meaning about 6 years ago, but relatively recently. A lot of organisms use different environments. Sometimes they're in one environment, sometimes they're in another. Think of plants where the seed can land. It can land in a shady spot or in a sunny spot with more or less water, things like that. Same with animals. They'll often pick large types of environments that they choose to use. An observation that quantitative geneticists have made over the years is that heritability, remember that measure of the relation between offspring and parents? That's often different in different environments. So if you raise up the same plants in a sunny environment or a shady environment, say, and you calculate heritability, it'll actually be different in those different environments. Well, selection's also likely to be different in those different environments, right? In other words, there's a correlation between heritability and selection. That goes into this term. And that turns out to have a big effect. And I'm not going to go through, for time, I'm not going to go through the calculations of it. But if you calculate a situation, if you look at a situation where, say, you have two habitats, and in one of them, the heritability is 0.6, and the other, the heritability is 0.4. Those are pretty close. Those are both in the ranges of heritabilities we often see in natural populations. And say you select in opposite directions. In one environment, in the environment where heritability is 0.4, you select for increased body size. In the environment where the heritability is 0.6, you select for decreased body size. Here's what happens. When you're in the environment with the high heritability, that selection for decreased body size causes a bigger change. So things get smaller by a lot. When they're in the other habitat, where they're selected for increased body size, they get bigger, but by less, because heritability is lower in that environment. What this does is it skews the outcome, and it turns out to skew it quite a lot. Made up a case. I'll try to draw it where we had a phenotypic trait here, and fitness. And for two different environments, I made up overlapping distributions or overlapping functions. It's kind of look like that. So for one environment, say the yellow environment, this is the optimal phenotype and fitness drops off away from it. In the white environment, this is the optimal phenotype and fitness drops off away from it. Now let's say that in the, say the heritabilities are different. Let's say the heritability in the yellow environment is about 0.6. And the heritability in the white environment is 0.4. I set this up so that if something uses both environments equally, the mean fitness on average across them actually has a single peak. like that. So if an organism's used with equal probability both environments, quantitative genetics, evolutionary game theory, adaptive dynamics would all predict that you should go to there. But if you force them to use both habitats equally, but they have different heritabilities like this, the actual equilibrium moves way over and it comes almost to this peak. In other words, you end up with a population that's quite well adapted to the yellow environment, very poorly adapted to the white environment. And the only difference was because the heritability was slightly higher in the yellow environment than in the white environment. So small differences in heritability turn out to be able to produce a large change in where the population ends up. And you end up with a case, if you force organisms to use both environments, individuals, half the time they're in the white environment and they've got very low fitness in that environment. They seem very maladapted to that. Now, in that case, I forced them to use 50-50. What if you let that change? So then I did a multivariate version. We don't have time to go into how to do the multivariate version of this, but I've solved that. Do the multivariate version where there's another trait, which is habitat preference. Which one do you prefer to use? Initially, there's no preference. However, as the population adapts to being good at the yellow environment, then that imposes selection for a preference. And so you end up with, initially you adapt to better to one environment than the other simply because of the different heritability. Then selection for habitat preference causes you to prefer to use that habitat. And so you end up with an organism that prefers to use this habitat and does well in it and prefers to not use that one. The difference is not that fitness is necessarily any higher in the yellow habitat than the other. In fact, you can make cases where it's lower, but heritability is higher. So this term illustrates new biology no one's ever studied before, the relationship between selection and heritability. In quantitative genetics, heritability is just treated as a number. You just stick it on there. In reality, it's a random variable. And when we allow it to be a random variable, that fact comes out. Does that make sense? How that works? Okay. Just quickly point out the other new thing here. And that other new thing is this, omega hat. Remember, omega is relative fitness, W over W bar. Omega hat, then, is the expected value of W over W bar. Omega hat is expected value W over W bar. But remember, this is probability expected value. These are both random variables. It turns out the expected value of a ratio of random variables, first of all, is not the ratio of expected values. This does not equal expected value of W over expected value of W bar. Okay, that would be convenient, but it's not the truth. In fact, it is a function of the entire distribution of W bar. And how to deal with this, the simplest way is to expand W over W bar as a series. I originally did that using Taylor's theorem, just expand it as a Taylor series. And you can do that in such a way that you get W bar in the numerator. Okay. There's also a problem with it. Mathematicians in the room know Taylor's theorem applies on a radius, right? Outside the radius, functions can become non-analytic, meaning Taylor's theorem doesn't apply. Most of the cases we run into in simple mathematical biology, the functions that we're using Taylor's theorem on are analytic everywhere, right? So things like e to the x or sine x, those are analytic everywhere. You can keep applying Taylor's theorem, you make the series longer and longer, you approximate it further and further out. Every calculus textbook has a picture with a sine wave, and they do like first order approximation, second, third, fourth, and they show it matching the sine wave going way out. 1 over something, 1 over W bar, becomes non-analytic at a certain point that is biologically perfectly reasonable. So, yeah, and if mathematicians are interested in this, I'll tell you why it happens afterwards. But as a consequence, Taylor's theorem doesn't really work for it. The way to solve it is to use orthogonal polynomials. And I'm not even going to write it up because we're just about done, and I'm not going to introduce a whole new topic for you here. But there is a way to take a random variable, any variable, and expand it as a series expansion in a population that always works. It doesn't run into the problems of Taylor's theorem. Furthermore, for a finite population, the series are always finite series. So they always converge in a finite period of time. And when you do that with omega hat, what you end up with is a series of terms that go out to higher and higher order. The next term after the expected value has the variance in fitness in it. And so when you expand this term, we actually end up with something that includes the covariance of phenotype, and actually you can break it up so it's just parent phenotype, with the probability variance in fitness. In other words, things are selected to, or not, you may call this selection, you may not call it selection. Things tend to evolve in such a way, and this has a minus sign in front of it, in such a way as to minimize fitness variance. And just to let you know that this happens, I'm just going to give an example, and you can play with this on your own. Imagine we have a case like this. A simple organism has two phenotypic values. Phenotype can be 0 or 1. They can be an allele, can be whatever. And I'm going to put individual fitness. This is W, not W bar over here. And it can have values 0, 1, or 2. And let's say that if it has phenotype 0, there's a 50% chance that it has fitness 0 and a 50% chance that it has fitness of 2. And if it has phenotype 1, it has fitness of 1 with probability 1. Okay. In this case, what's the regression of fitness on phenotype? It's 0, right? Because these two have the same expected fitness. This one has a higher variance than this one does. Now let's make up a simple case. Simple case of one individual here, one individual here. So one individual with phenotype 0, one individual with phenotype 1. There are two possible outcomes. That could happen. It could be that this one has fitness 2. Okay. In that case, the initial mean phenotype is 1/2. In the next generation, this has left 2 descendants. This has only left 1. So there are 2 individuals with phenotype 0 and only 1 with phenotype 1. So the mean phenotype, right, is now 1/3. It's gone down to 1/3 in this case because these left more offspring, right? Okay, that's the new mean phenotype. What's 1/3 minus 1/2? It's minus 1/6. So in this case, delta phi bar is minus 1/6. What about this other possibility? With 50% chance, these might leave no offspring. If they leave no offspring, if this individual does, and this individual leaves 1 offspring, then the initial mean phenotype was 1/2. After that occurs, there's only 1 individual. They have phenotype 1, so the mean phenotype's 1. So in this case, delta phi bar is equal to 1/2. So if this is probabilistic, whether this is 0 or 1, what's the expected change? Well, if it's 50% probability, it's the mean of 1/2 and minus 1/6. Okay, which is, first of all, a positive change. And it should come out to delta phi bar hat is equal to actually positive 1/6. In other words, when you work through it, and I did this exact case just to confirm it, sure enough, there's an expected change in this direction. Even though the mean fitness here is the same as the mean fitness here, the expected fitness of this individual is the same as the expected fitness of this individual. So this term that had phenotype with variance of fitness had a minus sign in front of it, meaning you tend to be pulled towards phenotypes with low variance in fitness. That actually happens in populations. And when you take this equation and you use these rules, especially using orthogonal polynomials to expand it, you end up with all sorts of new terms like this one, like this one. Each of those has a biological interpretation. Some of these are things people talked about before, some of them are not. But this provides a way to discover new evolutionary processes, starting out with this general rule that makes no assumptions, expanding it, and then examining these new terms that show up. Okay. So I give that as an example of, first of all, why stochastic processes, making evolution stochastic, doesn't just change the variance in the outcome of evolution, it actually changes the direction. There are directional forces. This is a directional force. This is a directional force. Both of these are. That are invisible in a deterministic model, but that become visible in a stochastic model. Furthermore, this stochastic model does not make any simplifying assumptions whatsoever about the shape of the distribution of those random variables. And so I want to give it to you guys who do biological modeling in a lot of different contexts. As an example, you can make something stochastic by adding a stochastic variable and assuming it's normal 0, 1. But you can also do it by just asserting that the variables you had now are random variables, and then using these rules that I presented for combining the stochastic, combining the probabilistic and the frequency operations. Okay, and we're out of time, so I'll stop there. Hope you found this set of lectures of value somewhat. There are a bunch of things, especially in the last part here, that I just kind of glossed over, including why 1 over W bar, when you expand it in a Taylor series, doesn't work. If you're interested in those things, let me know. I'll be around for a bit, and I'm happy to explain those. Otherwise, we'll stop there. And I'll tell these people they can bring their, you can bring coffee in if you want.
Yeah, the coffee's coming.
Yeah, okay.
I'll take the photo machine to take a photo.
Okay. Is that common in Latin American countries? It used to be in the United States, and then it stopped being. And I thought it was me. When I first got a faculty position, after I was a postdoc and I got a regular faculty position, people would do that at the end of the semester. And then they kind of just did it less, and then they stopped completely. And I thought, I'm getting worse at this. But then I talked to other people, and it stopped being a tradition, like people stopped doing it. And so it wasn't just me. Either that, or I am the cause of that no longer being a tradition. I hope that that's not what was going on, but anyway.
They are coming with the photo to take a photo.
Take a photo.
Yeah, yeah.
Okay, I'm supposed to be in the photo, I